Hello, this is Lab 9, where we're going to focus on nervous tissue, spinal cord, nerves, and reflexes. Now, as we move through this, keep in mind that we're going to have a subsequent lab that's going to focus purely on the brain, and a subsequent lab that focuses on the special senses. What you're going to notice here is the exercises 5.11, which is basically going to look at the microscopic anatomy. You're also going to have section 14.4 and 14.7, which is going to cover some of the aspects of the spinal cord, as well as 16 and 17. So let's get started. You are to review the exercises on 5.10, 14.4 through 14.7, 16, and 17. I will try to provide some images, but you are also to look at the images that are in your exercise book for the nervous system, from the nervous system slides, nerve and spinal cord models, reflex diagrams. I will be able, I am hopeful, of finding some very nice videos, which I encourage you to review for all lab videos. Some of them are going to be able to do a variety of the spinal cord models, nerve distribution, as well as the two-point discrimination test. Two-point discrimination test, by the way, can be done by yourself using something as simple as a paper clip that you space out at a certain distance and follow the instructions, and you'll be able to understand the sensory distribution and the sensory regions. Okay? Now, at the end, of course, you're going to use exercise 15 and 16, where you will basically fill out and you will upload in your Blackboard, all right, and make sure that they're saved as a PDF form. And these are due by Friday, 5 p.m. Let's get started. Now, if you look at 5.10, we have to be aware that nervous tissue is responsible for three basic functions: reception, integration, and the response. Reception is defined as with aid from the sensory receptors, we're getting input that's going to be heading toward the brain. It's going to detect changes either inside or outside of the body. The sensory information will travel in afferent pathways to the brain. At the brain, you're going to have integration. Now, I need to clarify one other thing. Some of this integration may not occur at the brain, but at the spinal cord. That's why we include both the spinal cord and the brain together, as we call it the central nervous system part. The integration is going to interpret and integrate sensory input, as well as store information in memory and producing thoughts. But the integration is going to also send out the response if one is needed, particularly when we see reflexes. The response, you're going to respond to that sensory input by either triggering muscle contractions or glandular secretions. You need to be aware of that there are two types of nervous tissue cells: neurons, which transfer and transmit action potentials, and glial cells, which are the supportive cells. The nerve cell consists of several important structures: the cell body, otherwise known as soma, and this is where you're going to have the location of the nucleus, the nucleolus, and in that area, you're going to have what is called missile bodies, which are really the endoplasmic reticulum, the rough endoplasmic reticulum particularly. The axon hillock is the funnel-shaped structure. This is the starting site for action potentials. The axon is the extension of the cell body and is the transmitting end of the cell. And of course, you're going to have terminal parts to that axon that transmit the information to either another cell, such as muscle or gland, or to another neuron. Dendrites are extensions near the cell body. They are the receiving end of the cell. Axons end at that synaptic knobs, we call them synaptic terminals or axonal terminals, to release neurotransmitter to the target cell. As I said, the target cell could be a gland, a muscle, or a neuron. Glial cells play a supportive roles in neurons, both in the central nervous system, CNS, as well as the peripheral nervous system, the PNS. Now, this concept map I produced before, and I encourage you earlier in the course to print it out. You can use this as a review tool. It's going to be very important. And it's basically like a dichotomous key. Ask you a question, you go down a certain pathway, whether it's yes or no. Does the cell produce action potentials? Yes, then it's a neuron. If it's no, then you start asking a subsequent series of questions. Does the cell form a myelin covering around the neurons? Yes. Is that cell present in the central nervous system? Yes, it's an oligodendrocyte. If it's no, it's a Schwann cell or satellite cells. Now, what if it doesn't have myelination around the neurons? Does it wrap around the brain capillaries? Yes, it would be an astrocyte. If it's no, then you ask yourself, does the cell act as an immune cell? If that's yes, it's a microglial cell. And if it is no, then it's a ependymal cell. Here is the structure that we were talking about a minute ago. You have the soma here. This is the cell body. You're going to see quite prominently the nucleus, and inside of the nucleus is the nucleolus. You will see some mitochondria here, here, and here. You will find them also down here as well. The dendrites are the receptive regions. They are going to be picking up either sensory input or input from another neuron, etc. This funnel-shaped structure at the edge of the cell body is called the axon hillock. Now, depending on who you read, they'll either say that the hillock itself is the generating site for action potentials or just slightly below that. Now, I want you to be aware of that because there is perhaps some developing data that may clarify this. But the axon we know is this very large extension, and it is the impulse generating and conducting region carrying along an action potential. But an axon potential on a raw, unmyelinated neuron goes only so fast. You want to speed it up? What you're going to do is create the conditions of saltatory conduction, where basically the only areas that are not myelinated are where you're going to have action potentials jump to jump to jump to jump, which means that the speed over the length or the distance of that action potential, of that axon is going to travel much faster to get to the terminal branches. How does this occur? Myelination is basically where you have, for example, Schwann cells or oligodendrocytes. If it's in the central nervous system, it's oligodendrocytes. If it's in the peripheral nervous system, they're called Schwann cells. They wrap around to form myelination. This myelination protects and sort of acts as an insulator for that length of the axon. Now, you might say to yourself, okay, so, well, the outermost covering is called the neurolemma. And think about this. This is basically taking, like, as some people would refer to as the jelly roll approach, wrapping cell membrane around and around and around and around. And what is cell membrane? Basically just phospholipids, glycolipids, things like that. So it's mostly acting as an insulator. And what you have are the impulses or the action potentials jumping. Instead of traveling all the way down this axon, they're jumping fast, 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 bang, 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 bang. And so the traveling speed, if you'll say, of an action potential is much greater with myelination. By the way, on a related concept, you've heard of multiple sclerosis. Multiple sclerosis is an autoimmune disease that somehow the body starts destroying these Schwann cells. Without the Schwann cells, you don't have myelination. Without the myelination, you may not have the transmission of the action potential, or as some people go into remission, you will have the function of action potential be much slower, which means that the signal is slower and thereby it may be weaker, or it's much more difficult to get down throughout the entire length of the axon. Also, let me just clarify something. The areas that are not myelinated are referred to as nodes of Ranvier, okay? And then we, of course, have our axon terminals down here. Here again is another example. And by the way, another term for cell body is perikaryon. In it, to maintain this unusual shape, as you can see here, I mean, it's not a round spherical shape. You have neural filaments. These filaments provide the cytoskeletal structure that basically allows it to take on this very unusual shape and to have certain extensions off of it. Nissl bodies are the clusters of rough endoplasmic reticulum and free ribosomes. As I said, you have mitochondria here. The axon hillock, and now a lot of times they're saying now the initial segment where the generation of action potentials is basically, how can I say it, is basically, it's proximal but inferior to the axon hillock. Now, the cytoplasm here in the axon is called the axoplasm. The cell membrane of this axon is called axolemma. And then we have branches here, which are referred to as collaterals or telodendria. And then we have these bulb-like structures that we call axon terminals. Now, this is where we get into 14.4 to 14.7. Let's talk about the spinal cord for a few minutes. The spinal cord consists of gray matter, which is neuron cell bodies. They're usually located more on the inside, and white matter, which is consisting of heavily myelinated axons, which is sort of on the outer edges of the spinal cord. The gray matter structures include the posterior horn, which receives sensory fibers from the spinal nerves, the lateral horn, which is present in the thoracic and lumbar spinal cord levels containing the cell bodies of autonomic nervous system. The gray commissure, which is a narrow band of tissue that connects the gray matter on each side of that spinal cord. The central canal, which is kind of in the middle, and it stands right actually dead center in the middle of the gray commissure. And this contains cerebral spinal fluid and passes through the gray commissure, as I said. And then we have the anterior horn, which contains cell bodies of somatic, that is voluntary motor neurons. And as you can see here, here's our gray commissure, here's our central canal right there. Posterior gray horn, lateral gray horn, anterior gray horn. Now, we are also going to have to be aware of the fact that you're going to have sensory information coming in and also motor output coming out. We're going to talk all about that. Sensory nuclei are referred to either whether they're somatic or visceral. When I say somatic, that's periphery of the body, your hand, the bottoms of your feet. If you step on a tack, boom. Visceral would be those things that you feel inside. Like, let's say you get a stomach ache, and you've had too many chili peppers or whatever, and you get that burning feeling. That's the sensory information from the visceral circuitry, okay? Now, when we talk about the motor nuclei, this is information leaving, going on efferent pathways out to the rest of your body. The visceral part, as you can see, is a little bit smaller because there's only so much that you can control. But notice that a lot of it is parallel to the lateral gray horn in the sense of that a lot of the visceral work is going to be autonomous, which basically means you're not in control of it a lot, okay? The somatic is much larger, taking up the space here of this gray matter, and that's going to be moving around conscious skeletal muscle contractions, moving your arms, moving your legs, things like that, okay? Now, as you move through this, we note that, oh, by the way, this is 242 and 243, not 2437. That's a typo there, sorry. White matter is divided into three distinct columns. They're called fununculi. The posterior fununculi is connected to the dorsal white column. The lateral fununculi is to the lateral white column, and the anterior fununculi is to the ventral white column. Now, what's going on here? You're sitting there going, I don't get it. What is all this wiring detail? Well, it's very important. When you look at ascending tracts from the part of the spinal cord, they're transmitting sensory information from the sensory receptors to the brain. When you're talking about descending tracts, they're transmitting motor information to the effectors. That could be muscles or glands. So basically, you have to have a means to take, let's say, when somebody drops something on your big toe, it's going to pass up through these nerves, up into the spinal cord, and then up through these fununculi to the brain so that you can go, ouch. You come and become conscious of the pressure and the pain and everything else, and you may also react. Now, the descending tracts are transmitting motor information to the effectors. For example, as soon as you go through this ouch, you may start also pulling up and looking at the toe and examining it, moving the toe away from whatever was that crushing pressure or that blow. And you may, of course, also been dancing on one foot, but not the foot where the toe was, but the other opposing toe. We'll get into more about reflexes, though, in a little bit. So here we have the different posterior gray horns, as you can see. And we have also, we talked about the central canal. This is all white matter here, 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 over here, etc. One other thing I wanted to bring to your attention is this. You're going to have also the meninges. Now, we talk about the meninges a lot in the brain, but they are existing in the spinal cord. One of the things you're going to see here is it's the similar path where you're going to have right intimately close to the spinal cord in contact with it is the pia mater. Then you have this arachnoid matter, which is outward in the middle. But unfortunately, with this microscopic slide and the way this sample was taken, the slice, it's not very clear. But what you can see clearly is the dura mater right here. And this all is absolutely essential because it's going to protect the spinal cord. Okay? Now, when we talk about in activity 14.5, the peripheral nervous system, PNS, this includes all nervous tissue outside of the CNS, the central nervous system, which is basically brain and spinal cord. In the PNS, each spinal nerve is formed by the union of the ventral root, that's the motor nerve fibers, and the dorsal root, that's the sensory nerve fibers. The dorsal root ganglion contains sensory fibers of unipolar neurons. Let me bring you back to this image right here for a minute. Okay, so on the bottom here, the anterior root, that's going to be bringing out motor information. This upper root and the posterior root ganglion, this is going to be processing and bringing into the spinal cord sensory information. But after they have merged and they've come to here, here and here, you can see are spinal nerves. Okay? So, a better image with the diagram helps you see this right now. To the anterior root, we have leaving, this is going to be motor information. Coming up through here is the sensory information and eventually goes into the posterior root. Okay? Right around here, they fused, and basically the axons are carrying information. They're either carrying it sensory to the afferent pathways back into the spinal cord, or they're taking information and leaving the spinal cord on efferent pathways to certain motor responses, either spinal, either muscles or glands. Okay? Now, in activity 14.6, we note that the peripheral nerve, either cranial or spinal nerves, will contain thousands of nerve fibers. Each nerve fiber, the axon, is bundled into groups called fascicles and is enclosed in connective tissue coverings. You want to keep in mind that a nerve itself will also have some blood vessels inside of it. So what makes up the nerve is a connective tissue covering, a connective couple of layers of that. You're going to have lots and lots of these axon fibers passing through, and you may have some blood vessels present too. Now, some fibers are also going to have the myelin sheaths. So we have to get familiarized with these coverings and these protective sheaths that exist for the entire nerve. These include the epineurium, perineurium, and endoneurium. The epineurium is a layer of connective tissue that surrounds the entire nerve. It's sort of the outside. And that's when we start getting, once we get inside, these bundles that are called nerve fascicles. So it's a bundle of these fibers. These are surrounded by the perineurium. And then finally, for individual ones, you're going to have the connective tissue sheath that surrounds the individual nerve fibers, and that's the endoneurium. Here you can see in this diagram, we have it all. The epineurium, in these fascicles, they have the perineurium here in the purple, okay? And then you have the individual coverings, which are the endoneurium. By the way, notice through some of this connective tissue, you've got arteries and veins here and here and here and here. Now, also one thing to keep in mind, if this was a myelinated axon, the endoneurium wraps around the outer part here so that you can have sufficient Schwann cells producing the myelination that you have here, because that is your actual axon right there. In activity 14.7, we talk about the myelin sheath further. This consists of fused layers of cell membrane from special neuroglial cells. The high lipid content provides an insulating layer for the nerve fibers. Now, in the PNS, the peripheral nervous system, these are done by Schwann cells. In the CNS, this is done by oligodendrocytes. The axons surrounded by myelin are called myelinated fibers or myelinated axons. They may have internodes, site of myelination, on the axon fiber. The sites that are not myelinated are basically these little gaps that are called nodes of Ranvier. They're regular gaps of myelination along the axon, and this is so we can have the action potential, which henceforth will probably periodically use AP for abbreviation. So the APs travel faster in myelinated axons than unmyelinated. That's why when you have something like multiple sclerosis and you start destroying myelination, the capability to carry speedy information from one neuron to the next is basically reduced. Action potentials may still pass, and usually it's demonstrated when you have remission, but during this time, you also will have some of this tissue scleric or becoming hardened tissue, but you won't get the same speed of action potential travel along that same axon because of the loss of myelination. If we have an action potential passage on unmyelinated axons, this is called continuous propagation. If we have action potentials pass on myelinated axon, and this is where the action potential skips from node of Ranvier to node of Ranvier to node of Ranvier. This is called saltatory propagation. Here again is the neural limit. Okay, so we have this myelination here. This is the Schwann cell, and if you notice close, you'll see how the myelin has been basically being wrapped continuously. And so what happens is most of the cytoplasm, etc., of this Schwann cell gets squeezed out. So all you really have are basically layers of cell membrane until you get to the outside here. This is where you're going to have the nucleus, by the way. So this is all myelinated. It's the internode. Here, every place that's not myelinated is known as the node of Ranvier. Okay? You're not going to get myelination at the initial segment that's unmyelinated. Here's the axon hillock. Here's the nucleus, the cell body. Okay? Now, let's get some clarity here also. When we look at neuroglia at the central nervous system, We have ependymal cells, which are going to line the ventricles in the brain. Okay. We have microglia, which actually acts somewhat like an immune system. It will go in and kill and basically remove any dead tissue or dead material. That's the little guy right here. They kind of call them spiny looking cells. Okay. As a matter of fact, if a section of the brain dies, the microglia will migrate in and they will repopulate the area after they have phagocytized all of the dead tissue material. You don't get recovery and regeneration of neurons. Astrocytes are a little different. They have these extensions, pseudopodia, that basically wrap around capillaries. And then they also wrap around neurons, particularly on the axonal area. Let's get to the axons that actually have the white matter. Gray matter, remember, is just unmyelinated, and it's cell bodies for the most part. White matter is because you're going to have lots of myelinated axons. Notice that you have this oligodendrocyte, which will send off very large sections of its cytoplasm, and it will wrap around the axons. And notice that you can have an oligodendrocyte actually cover or protect several cells at the same time. That's not the same as what you would have with a Schwann cell. One cell, one wrapping, that's it, for just one axon. Now we're going to move into spinal meninges and the spinal cord. So let's talk about this. This is activity 16.1. Now, the spinal cord is protected by the same three meningeal layers as the brain. I talked about that earlier. You have the dura mater, which is the toughest outer layer. The arachnoid mater, which is the middle layer for spacing for passage of the cerebral spinal fluid, CSF. And then you have the pia mater, which is a thin layer in contact with the spinal cord. Now, the layers are basically continuous with the cranial meninges at the foramen magnum of the skull. You remember that's, if you look underneath the skull, you get that big hole area. That's where the spinal cord goes in and meets up with the brain. Then you have the subarachnoid space. This is space underneath the arachnoid mater, and it's filled with cerebral spinal fluid, CSF. You also have what are called denculate ligaments. Now, let me explain to you denculate ligaments. The spinal cord does not fill from edge to edge the vertebral foramen that exists on each individual vertebrae. There may actually be a small gap of space. What we want to do is prevent lateral shifting from one side to the other of the spinal cord. Doesn't need to bang around inside of your vertebrae. So we have lateral extensions of the pia mater, which connect to the spinal cord, and they connect further to the dura mater, and they protect the spinal cord from movement inside of the vertebral space. And I'll show you actually cadaver images of that in a minute. Here we have our spinal cord. We have our gray matter here, our white matter here, the anterior horn here, the posterior root here. So we have eventually the spinal nerve. Then we look at this further and we see the pia mater, that's intimately right attached to the spinal cord tissue. We have the arachnoid mater, which is next. And then we have the dura mater. Now, if you look further, you will see several other structures here. Okay, we have the subarachnoid space, which is right here. So this is the arachnoid meninges. This is the dura meninges. Now you'll notice also in that spacing of the vertebral space that there's going to be some padding here of adipose tissue here and down here. All right, so the epidural space right here is sometimes used for an area where you might have an injection. This is particularly true when you get down to the lumbar region of the vertebrae, because the spinal cord basically reduces itself in size and then eventually just becomes a large number of smaller spinal nerves. Okay? You have the meningeal membranes continuous with the spinal nerve connective tissue here. Okay? This would be, of course, the final spinal nerve here. What's this here and here? We'll show you. That is the tissue I was talking about, the dentulate ligaments. So basically they have attached, and they're serious anchoring that allows no lateral movement side to side, bang, bang, bang. You don't want that, because that any contact would cause trauma. Trauma may interfere with the normal function of the spinal cord. Here's the pia mater, okay? You can see the posterior root here. You can see the anterior root here. You can see the arachnoid matter, which is cut and reflected, meaning it's pulled back a bit. And then underneath, and above this, I should say, is the dura mater, and it's somewhat cut and reflected. Okay, so here's our spinal cord. And you'll notice also some blood vessels within the subarachnoid space right up through here. Now, the subarachnoid space extends beyond the end of the spinal cord. This is where we're going to get into what I was mentioning earlier, when we get into the lumbar region. We have a structure called the conus medullaris. It's a cone-shaped termination of the spinal cord. It ends somewhere between vertebrae L1 and L2. The dura mater and the arachnoid mater extend beyond the conus medullaris to between S2 and S3. The subarachnoid space ends as a blind sac, and it's filled with cerebral spinal fluid. This entire area is very useful for what we call the epidural block. That's an injection site for anesthetics, and lumbar puncture, which you can use to make a spinal tap. In other words, you take some sample of the cerebral spinal fluid. This is fantastic because what you're looking at is fluid that looks somewhat like water, if it's healthy, okay? But it's going to contain proteins. It will indicate to you if you have the presence of bacterial or viral infection. It will indicate for you the ion content of the cerebral spinal fluid. So it's a great diagnostic resource. Now, there is a structure you also need to be aware of, and that's called the filium terminae. That's a thin strand of pia mater that extends from the conus medullaris and extends all the way down to the coccyx, sort of like an anchoring structure. So you see where the flow of cerebral spinal fluid occurs throughout the system, and then it will go down. It goes around the spinal cord. It will go down through the central canal of the spinal cord, and it flows through this also the subarachnoid space that you see around the spinal cord. You have the dura mater here. Okay, so underneath you got the arachnoid, now you've got the subarachnoid space, and now you come down to where the cauda equa is, and you just have a lot of nerves, and you eventually have the filium terminae. That's that structure you can see right here. So you need to keep in mind something. Once you get about L1, L2, there's a perfect area right there that you can go in between the vertebrae and send a needle in. And I've seen this in surgery, and the doctor will take out some fluid for a little bit, and then instead he will inject in replacement the anesthetic, and this will give you the epidural block, so everything in the sense of feeling from that point downward, particularly pain, will be silenced. All right? Now, let's talk about the origin of the spinal cord at the foramen magnum on the base of the skull. This is protected by the vertebral canal. The spinal cord is divided into 31 segments. Cervical is 8, that's C1 through C7. Thoracic is 12, T1 through T12. Lumbar is 5, that's L1 through L5. And sacral, which is 5, which is S1 through S5. And then we have a designation slightly different. Be aware of that. The coccygeal, there's only one, and it's labeled CO1. There are 2 enlargements of the spinal cords. The reason we have these enlargements or thickenings is because it's got to manage the nerves from the limbs. So up in the cervical area, there'll be this sort of widening there, and that's for the nerves coming up, bringing information from the upper limbs. The lumbar has a thickening area there, and that is for handling the nervous input and output for the lower limbs. And you can see this right here. This is the cervical enlargement right here. Here, down here, is the lumbosacral enlargement right around here. Okay. You have the filium terminae, which comes all the way down to the coxogenial area. It's basically the anchor to prevent the nerves here. You're going to have the cauda equina, and basically what that means is like horsetail. And what's going on is, instead of this being one solid massive spinal cord, it now basically subjugates itself into just a series of spinal nerves. And these will pass out in the various areas of the vertebrae all the way down, and then they have very long axons to various lower body extremities. Okay, keep in mind, of course, here we have the cervical spinal nerves, the thoracic spinal nerves, the lumbar spinal nerves, the sacral spinal nerves, and the coxogenial. And I think I said, yeah, 8. They come out between C1 and C7, but there's one that comes out from around a little bit above the C8. So what you've got is always coming above or out on the sides. Okay? So we have the posterior medial sulcus right here, and the inferior tip of the spinal cord's right there, and then we have the cauda equina. All right? And you'll notice also the curvatures. Here's what I wanted you to get earlier. See, as we end up getting down to the conus medullaris, you see all these nerves still coming out. Now, this is the conus medullaris, all of these finer nerves that will come out in all of these areas. Okay? When we talk about spinal nerves, they're always mixed nerves, as they contain sensory input, which would be up afferent pathways, and motor output, which would go down through efferent pathways for nerve fibers. There are 3 main branches. In this case, we call them ramus or rami. Ramus for singular, rami, of the spinal nerves. There's the dorsal primary ramus, which basically supplies the deep back muscles and the skin of the dorsal surface of the trunk. The ventral primary ramus, which supplies the structures on the limbs, the skin along the lateral and ventral surfaces of the trunk, and the skeletal muscles except the deep back muscles. Remember that that's taken care of by the dorsal primary ramus. Then we also have a structure called the rami communicantes. These are branches along the thoracic and lumbar spinal nerves. They're associated with the sympathetic division of the autonomic nervous system. So remember, here's where we have the anterior ramus here, the posterior ramus here, and the rami communicantes, because what happens is the pathway coming out here will first go through the sympathetic ganglion. You will have an autonomic nerve come out, and then it will go back and, if necessary, follow down. When we talk about the spinal cord further, you need to understand that in some cases, it is not just one nerve going all the way down to your toes, going all the way back up to your brain. It may be a chain. This is where we get into the first, second, and third order neurons. White matter in the spinal cord contains ascending sensory pathways and descending motor pathways. We've already talked about that. Now, ascending pathways in the spinal cord transmit general sensation to the primary sensory area in the post-central gyrus of the parietal lobe of the brain. We'll get more into that next week, but there is a section in the brain that takes up all the general sensation. When I'm talking general, I mean things like, okay, a feather brushed up against me, I just had a horsefly bite me, or a mosquito bit me, and now I'm itchy at that spot. That's sensory input. There is what we call a sensory homunculus. It is a body parts map that compares the relative size of the sensation areas in the brain. So in this particular area of the primary sensory cortex in the postcentral gyrus, you can map out There's going to be lots of neurons that are processing this information from hands and parts of the face, and not as much from legs or elbows or upper arms or chest. Okay? But let's talk about how this chain of information passes. The first order neurons transmit sensory information from a receptor to the spinal cord or brainstem to be passed on to the second order of neurons. The second order of neurons cross over to the opposite side and travel to the thalamus to synapse on a third order neuron. Now, in the thalamus, you have to understand it's a section of the brain that's going to pass information to the respective lobes of the brain that process this type of sensory information. The third order neurons travel from the thalamus to the cerebral cortex. When we talk about a receptive field, this is an area of the body monitored by the single sensory neuron. This is where we can find out how much and how dense is the receptive field. In other words, can you really sense a lot in a very small area, or will you get a larger area that a single neuron is covering? This is where we get into a two-point discrimination test. As I said, you can unfold a paperclip and don't pierce your skin, but basically just touch on it. You can assess the spinal nerve function and compare the relative density of receptors in the skin of a selected body region. Here's the first, second, third order. I'll get into this in a second. The first order brings up the sensory information to the spinal cord. The second order passes it over to the opposite side. Remember, we always say that it's the opposite side of the brain that controls one side of the body. So your left side is actually controlled by the right side of the brain. Your left arm, right brain. Right arm, left brain. So here we have the pathway. We've gotten the first order neuron. The second order neuron gets up to the thalamus. It synapses with a third order neuron, which then passes it over to the primary somatosensory cortex. This strange looking caricature here is the homunculus. And what you're saying basically is the amount of cortex determines how much the receptive fields and the sensory systems are. As you can see, there's a large amount in the hand. A much smaller amount is given to the arm. A modest amount to the chest, etc. And then you get into the foot, which has got a lot more. And even when you do the face, you look at there's a sizable amount of cortex that processes the lips and just the general facial region. Okay? Now let's do receptive area. Here's the thing. If you have a receptive field that's right here, you have two probes touching here, you would get the same detected sensation. So you wouldn't be able to tell if you have one or two probes there. But if you get the prongs separated sufficiently on a receptive area and you have one hit this field and one hit that field, you're going to note that there's the two prongs that you've come in contact with. Now we have to talk about the sensory cutaneous regions of the body. This is where we get into dermatomes. These are differences of the sensory cutaneous regions of the body innervated by a single pair of spinal nerves. Adjacent dermatomes on the trunk will have some overlap, and variations of individuals does occur. The peripheral nerves are derived from the brachial plexus. They will be used as a model for the testing of functions of the individual spinal nerves. The brachial plexus is a network, a plexus, of nerves formed by the ventral ramus of the lower four cervical nerves and the first thoracic nerve. So C5, C6, C7, C8, and T1, they fuse together and then form this network. And then this plexus extends from the spinal cord through the cervicoaxillary canal in the neck, over the first rib, and into the armpit. If you want to do some of the testing that you see on page 287, it'll help you to understand where some of these things are. For example, if you take a look just at the hand, various spinal cord regions play a role in the detection of this. Okay? Why is this important? Patient A comes in and has had a spinal cord cessation right here between T1 and C8. If you wanted to map out where the injury has caused damage, so you've lost sensation, you can map it out even with the hand, because C8 might be detected, T1 would not. So if you tap on this finger, I don't feel a thing. Tap on this, I feel something. Tap on this one, it's C7, I feel something. Tap on the thumb, which is coming in from C6, I feel something. That's just one point that I'm trying to bring up. And if you notice, this is anterior side, this is posterior side. So you can see how we move from the cervical to the thoracic, and then as we get lower, you have a lot more on the back side for the lumbar here. The back side for the lumbar on the front is taken up over here. And then we have the sacral, which is all taken up in the back part of the leg, etc. You can see this also of specific nerves, okay? Obturator femoral, genital femoral, ilioinguinal, the radial ulnar, medial nerve, and iliohypogastric nerve, etc. Do I need to know these? Yes. Okay? Now, we're going to move into activity 17. The cerebellum receives the following inputs. The motor commands from the cerebellum. The stimuli for equilibrium, that is balance, from the inner ear. Stimuli for vision from the eyes, and stimuli from proprioception. This is the body position in space. In other words, are your arms up? Are they down? Are your lower forearms folded up, or are they totally extended downward? This stimuli is based on what they call proprioceptors in muscle and joints. The cerebellum sends information back to the cerebrum to fine-tune the motor activity. The normal cerebellar processing allows for performing dancing, walking, balancing, and climbing a ladder. You see, what I'm trying to help you to understand here is this. The cerebellum, even though it's considered the little brain, is really key to coordination, balance, to also have measured responses in how you extend your limbs and how you move them instead of a staticky, choppy movement, but a nice smooth flow. And this is all based on interactions from sensory input processed through the cerebellum and then given, processed to the cerebellum and then given to the cerebrum to fine tune and develop those smooth movements, those coordinated movements. I encourage you to review the pages 292 to 297 to perform tests on coordination, movement, and balance. You don't have to do that as much, but you know what? If you've got either someone that you know or you want to do it yourself, you can do some of these. Be careful. Never do them if you're sick. Never do them if you are injured or have an inability to manage your coordination, movement, or balance. I always say the same thing in labs. One of the other things is that do this with the sense that you can stop at any time, okay? If it gets too much, stop it. Record the information in your lab book. You're not going to send it to me, but it would be helpful for you to understand some of these points. Now, if we look at this, we have here the cerebellum. There's sections of the midbrain that are up here, the pons, the medulla oblongata, and of course the spinal cord. But this information is passed up through here, okay? Now, you have the choroid plexus here, the fourth ventricle, and then you have the white matter that does the connections. The arbor vitae is literally just whitish. It's called the tree of life. That's what arbor vitae means. And what you're having is a lot of processing that goes into the cells of the cerebellum and then back out, and especially back up the brain. The gray matter, considered the cerebellar cortex and the cerebellar nuclei, the centerpiece here. And of course, you have these attachment structures called cerebellar peduncles, which are attachments and connect up to, from the midbrain here, to the cerebellum. And this includes the superior, the middle, and the inferior. Now, to perform reflex tests on the upper limb, you would record your results. One of the simple ways to do it is, some people would take a ruler, hold the upper part, and then have their hand over the lower part. And basically, as you let go of the ruler, you clasp it. Now, usually you can do that with someone else. They're holding it, you're going to clasp it. That's a reaction timing test, particularly for the reflexes. What's a reflex? A reflex is an automatic, involuntary response to a change that occurs inside or outside the body. Everybody knows, the doctor comes in, takes a little rubber mallet, pops on your patellar tendon, and your leg kicks. That's a reflex. They're checking, is the reflex normal? Because a reflex arc is a neural pathway of a reflex. It consists of the sensory receptor, the sensory neuron, the integration center, which, hint, hint, it should be either the spinal cord or the brain, or both, plays a role, the motor neuron, and the effector organ, which can be a gland or a muscle. Here we have a very simplified diagram showing it. Stimulus, poke your finger. Sensory receptors report this. Then this information is passed by sensory neurons up through here to eventually the dorsal root ganglia to the sensory neurons in the spinal cord. Now there's going to be collaterals from this. Some that are going to allow information to be passed up to the sensory processing centers of the brain. Others are going to have a direct input to a neighboring motor neuron here. And this will pass out of the CNS down through the PNS and cause it to be innervated at, let's say, the biceps muscle, which would cause a response. They pull away from whatever it was that was painful. To perform reflex tests on the upper limb, and you want to record your results, you can do several different tests. If you're at home and you're doing this, no major importance. If you can record the information, if you can do it at home, fine. If not, do not worry. But you need to be aware of what these are. Excuse me, what these reflex tests are. Stretch reflex, spinal reflexes are involving muscle spindles. These are receptors embedded between skeletal muscle fascicles. Now, upon stretching the muscle lengthens. Upon stretching, this stretching will trigger a reflex muscle contraction to prevent excessive stretching of the muscle. Now, the biceps reflex test assesses the function of C5 and C6 segments of the spinal cord. The triceps reflex test assesses the function of C7 and C8 segments of the spinal cord. And you can see some of this. Here's the biceps test. You're tapping down right around here at the tendon. The triceps, notice you have the forearm here, and you tap it around the side here, and you're going to get some response. Notice that you can tell from the arrow here what the response would be. In this case, you have the extension of the forearm. In this case, by tapping it, you would have basically a flexion of the forearm. The ankle jerk, if you tap on the Achilles tendon, you're going to have the foot go downward, and this is known as the ankle jerk reflex. Now, you might take this for granted, but you think about it, this is a great way to detect things like neurological damage, whether there is a disease that's causing neurological impairment right now, by just these simple, non-invasive tests. When you look at pages 300 to 301, they give you the information to perform the reflex tests on the lower limb. If you cannot do them yourself, you should absolutely know what they are. The patellar reflex test. This evaluates the function of L2, L3, and L4 levels of the spinal cord. The ankle jerk test, and this affects the Achilles as a reflex test. It evaluates the function of S1 and S2 levels of the spinal cord. The plantar reflex test, which is referred to also as the negative Babinski reflex. The normal response is the plantar flexion of the foot, and you examine the function of S1 and S2, and to a lesser extent L4, L5 segments of the spinal cord. Now, let me explain to you a little bit of how this is related to Babinski sign. In infants, because of the delay in myelination, what you're going to have is, with the plantar reflex test, you're going to have it opposite. So you're going to have the spreading of toes at the plantar test. This occurs until age 2. In older children and in adults, positive Babinski is the sign of damage to the descending motor tracts of the spinal cord. What you would have instead is a negative Babinski. Here's a Babinski reflex. You run the blunt part of the hammer up through here. If the toes spread and the child is, let's say, 1 year old, okay, that's normal. Normally what would happen is, with adults, if you run this up this way, the toes kind of curl. That's a reflex. But if somebody, I've been talking about problems with health and everything else, they do a Babinski reflex and the toes all flail out. That's the indication that lower motor neurons have been influenced, damaged, or affected. The plantar reflex, if you run that low amount through sort of the center of the plantar part of the foot, otherwise known as the sole of the foot, and you still get a curling. Okay? So we've pretty much covered everything. So what do you need to do? There are 2 sections that you need to fill out from worksheets and submit before 5 p.m. on Friday. This is for exercise 16 and 17. Parts of 14 and part 5.10, this is for your own review and edification. I do encourage you to take time to review the slide images because they're fair game for the second lab practical exam. The same images also to look at on 14.4 through 14.7, as well as 16 and 17. You want to review the nervous system slide images, spinal cord models, as well as the dermatome diagrams. Eventually, you are going to submit those documents for grading, and that's fine. I do encourage you to review the lab videos and the lab manual notes. I have included in those videos some short clinical videos that include things like the plantar test, the Babinski sign, etc. Well, have a nice week.